From splicing DNA to curing epidemics, the most inventive of doctors have radically improved our lives and kept us safe. Doctors Herbert Boyer and Stanley Cohen gave birth to an entirely new science when they became the first to perform genetic engineering. As an assistant professor at the University of California at San Francisco, Boyer observed that an enzyme could cut DNA strands in a very specific and particular fashion. Dr. Stanley Cohen, hailing from Brooklyn, New York, had been working at Stanford University, also studying the complexities of DNA. He was able to extract plasmids and transplant them into other cells. When these two medical pioneers met at a conference in Hawaii, they made history while discussing each other's discoveries over a deli sandwich. They combined their knowledge and together were able to invent a method of removing the DNA from one living organism and splicing it into another. They had unlocked the hidden secrets of DNA and invented what we now call genetic engineering. Boyer and Cohen viewed their revolutionary technology as a way to overcome disease or to increase food production. The beneficial results could be endless. New drugs have been made using Boyer and Cohen's science, including growth hormones and new cancer treatments. But along with great celebration comes cause for concern. In the wrong hands, genetic engineering can be used to make biological weapons, developing super viruses against which we have no defense. Genetic engineering also paved the road for a scientific advancement that has sparked great ethical debate: cloning. In February of 1997, it was announced that a sheep, affectionately named Dolly, was the first mammal successfully cloned, an exact copy of an adult sheep. The debates continue, but the science marches forward as new advances are made with DNA and genetically engineered cells. Since their discovery, Herbert Boyer has partnered up with venture capitalist Robert Swanson to form Genentech, one of the world's largest biotechnology companies. Stanley Cohen continues as a professor of genetics and medicine at Stanford University in California. It's amazing what we can accomplish today on a cellular level. And it all started over lunch between two of the greatest minds in science. A bacterial infection was once an immediate cause for concern and was a possible death sentence. It was the genius and quick thinking of Alexander Fleming who, by discovering penicillin, found a way to fight bacteria. Fleming was born and raised in Scotland until the age of 14, and then moved to London where he studied medicine. Although he initially wanted to be a surgeon, he ended up in the inoculation service. When World War I broke out, Fleming and his colleagues were sent to France to treat injured soldiers. He learned firsthand the dangers of infection on the battlefield. In 1928, Fleming stumbled upon a solution while studying bacteria in one of his petri dishes. He noticed a mold that was curiously killing the surrounding bacteria. Others before him had disregarded such findings, citing their experiment as contaminated. But Fleming's inventive mind had realized an opportunity. Fleming had inadvertently discovered penicillin and the potential it had to save lives. Fleming presented his findings in 1929. But it wasn't until World War II that penicillin would reach its true potential. The Allied and Axis powers fought on land, sea, and air with new, powerful, and more deadly weapons. Every day, more soldiers were injured and in danger of dying from infection. Australian-born Dr. Howard Florey and German émigré Dr. Ernst Chain picked up where Fleming left off and were able to isolate and mass produce penicillin. By 1944, there was enough penicillin to treat all wounded soldiers of the Allied armies. After the war, penicillin was used widely to treat all forms of infection and disease, earning pioneer medics Alexander Fleming, Howard Florey, and Ernst Chain the Nobel Prize in 1945. World War II heralded many medical innovations, but one of the most important to the success and livelihood of the Allied troops was the creation of blood banks, pioneered by Dr. Charles Drew. Born in Washington, D.C. in 1904, Dr. Drew excelled in science and medicine during a time when African Americans had very little opportunities for education and advancement. Fighting prejudice and overcoming poverty every step of the way, Dr. Drew eventually received his doctorate from Columbia University. He researched plasma, the liquid portion of blood that can be separated from whole blood. In emergencies, plasma transfusions help maintain blood pressure and volume and supply critical proteins for blood clotting and immunity. He developed a radically new way to house plasma in what he called blood banks. As England prepared for World War II, they realized the need for blood transfusions for their troops and turned to Dr. Drew and his innovations for help. He started the Blood for Britain campaign in London. As a result of his great success in England, the American Red Cross selected Dr. Drew to create and direct the Red Cross Blood Bank as the U.S. entered the war. The Red Cross collected almost 13 million units of blood and sent dried blood plasma to Allied troops fighting in Europe, the South Pacific, and North Africa. Dr. Drew's methods of collecting and storing plasma saved countless lives. But Dr. Drew became incensed when the U.S. Army requested that white blood and African American blood be segregated in transfusions. He knew that there was no scientific difference between blood of different races. Dr. Drew resigned from the Red Cross, partly due to the controversy. The government eventually admitted their error and ended the practice of segregating blood. Despite the dispute, Dr. Drew received many accolades for his life-saving contributions. He became the first African-American surgeon appointed to the American Board of Surgery. Tragically, in 1950, Dr. Drew died in an automobile accident, but his groundbreaking work in blood storage and transfusion and importance in promoting racial equality are everlasting. In the late 1940s and 1950s, every parent in America shared the same fear. Their child may contract polio, a crippling disease that caused permanent paralysis and chronic shortness of breath that often led to death. The country was all too familiar with the disease. U.S. President Franklin D. Roosevelt was a survivor who was confined to a wheelchair because of its effects. Polio was known to mostly affect children, so parents around the world were holding their breath for someone to create a vaccine. Dr. Jonas Salk answered that call. Salk was a very determined individual, a man with as much ambition as brains. After receiving his doctorate, he worked with the United States Army during World War II, creating flu vaccines for the soldiers overseas. After the war, he worked at the University of Pittsburgh, revamping the virus research laboratory, where he came to the attention of Basil O'Connor, director of the Foundation for Infantile Paralysis, later known as the March of Dimes. O'Connor immediately recruited Salk, and he gathered all the resources and funding needed to hopefully find a cure for polio. Salk plunged into his project, working to end this epidemic. he labored at an incredibly fast pace, as opposed to the traditional slow and methodical speed that had been a foundation in scientific research. Salk clashed with other researchers also for his presence in the media. He seemingly appeared in front of the camera almost as often as in the lab. Despite his differences with his colleagues, Salk worked diligently, and by April of 1954, he was ready for a massive field trial of 400,000 children. On April 12th of 1955, Salk's vaccine was pronounced effective. Salk was immediately claimed a national hero, known as the man who saved the children. Families immediately made appointments with their doctor for the miracle vaccination shot. But while the nation adored him, the scientific community felt very slighted. Salk claimed all the credit for himself and did not offer any gestures of gratitude or debt owed to those who came before him. Most upset was probably Albert Sabin, a virologist who was working on a different yet equally effective polio vaccine. But his vaccine was not approved until 1960, 5 years after Salk's. Despite internal gripes, all could breathe a sigh of relief. Dr. Jonas Salk may not have been typical, but he was definitely effective. Salk's vaccine rid the earth of polio, making the world just a little bit safer.